Occasionally in nature, you get two groups that have separated geographically or otherwise and have diverged and become what we consider different species. But then, as time and geography change, they come back together and they have at least a zone where the two groups can interact with one another. And occasionally that means they can produce hybrids, right? You can have individuals of both species mating with one another and producing hybrids of the two species. For example, here are two different species of toad in the genus Bombina. So we have the yellow-bellied toad and the fire-bellied toad that exist in sort of Western and Eastern Europe and Eurasia here. And they have this long, narrow area in the middle where they meet, and they have hybrids that develop there. Now, if you have a hybrid zone like we have with the genus Bombina, with our fire-bellied toads and our yellow-bellied toads, there are three possible outcomes when you have that mating. You can have reinforcement, fusion, and stability, and we'll talk about those one by one here. So we start off with a single population or single species, and you develop some sort of barrier to gene flow, geographic or otherwise, and then your isolated populations diverge from one another. Or you may have one main population and one isolated population. But eventually, you come back around and these populations are diverged enough that they're different, but they haven't diverged so much that you can't exchange DNA and mate and create hybrid individuals. But again, what happens to those hybrid individuals depends upon their fitness. Are they more fit or less fit than their parental species that they came from? In reinforcement, the hybrids are significantly less fit than their parent species. And over time, because if you mate with someone of the opposite species, your offspring are less fit. That means your genes don't get passed on. And so there's a lot of selection pressure to not mate with individuals of the opposite species, even in that hybrid zone, really especially in that hybrid zone. And so it ends up pushing those species apart. It reinforces the diversion between those two species. And here's an example. So this is a flycatcher, this type of bird. And if you take a female and you give them the choice of two males, one of their own species and one of the other species that are from a different area, right? They're not from that hybrid zone. They are going to more often choose their own species than the other species, right? They've selected to do that. But they're still going to choose the other species a fair amount of the time, which presumably will produce offspring that are then less fit, which is not good for that female. In the hybrid zone, so a sympatric male, meaning in the same geographical area, if females are choosing between sympatric males, they are always going to choose their own species, right? This shows you that there is a lot more selective pressure in those hybrid zones, right? When you're living next to the other species, to choose your own species, if in fact the hybrid offspring are less fit. Now, the hybrid offspring are not always less fit. Sometimes the hybrids are just as fit as their parents, in which case you have reestablished gene flow between those two species in that hybrid zone, and there's no real barrier to gene flow, right? And in this case, you may have fusion again. So instead of reinforcing that divergence between the two species, these two just barely different species that are pretty similar to one another may actually fuse and become one species again. So one example of this is some cichlids in Lake Victoria that we talked about before. So we already mentioned these guys, and that experiments showed that it was the color of these individuals, it was sexual selection that was causing them to diverge. But in an experimental monochromatic sort of orange light situation where you couldn't tell the difference between those two individuals by color, they would then mate with one another and exchange DNA. Well, now, because of some turbid water in a particular area, meaning it's not particularly clear, so some of this is a pollution issue, you do get that monochromatic light, and the colors can't be discerned quite as easily, and you are getting a hybrid individual, a hybrid species, I should say. It's popping up between these two species. But really what that's doing is it's creating gene flow, and it's fusing these two back into a single species. So we've talked about how if hybrid offspring are significantly less fit, then you get reinforcement of that division, right? And it further selects for those two species to continue diverging and to become sort of permanently different species. We also talked about how if those offspring are not less fit, then you get fusion, and those two species join back together into a single species because we've established gene flow. The third option is some sort of in between that we call stability. In this case, the hybrids are less fit, but they're not that less fit. And you have continuous gene flow coming in from outside the hybrid zone, so that you end up sort of just maintaining a status quo. So there are a lot of different ways that speciation can occur. And sometimes they can occur rapidly, or sometimes they can occur slowly over a lot of time. Sometimes speciation can occur because of just a few genes, or even a single gene, like we talked about with those left-handed and right-handed snails, or sometimes it takes lots of genes to diverge to become different species. Scientists who study the very broad patterns of speciation over huge amounts of geological time using the fossil record, morphological data, molecular data, there are lots of different ways to do it. They find a few different things. Sometimes you find in the fossil record what look like long periods of time where you have very little seeming change, and some periods of time where you have periods of drastic change. These were described by Niles Eldredge and Stephen Jay Gould as punctuated equilibria, these periods of apparent stasis followed by sudden change. And this is contrasted by a gradual change model, which we can see here, right, where we have a little bit of change at a time consistently over a long period of time. Now, it turns out that both of these can be the case. Different species follow different paths, and sometimes things look more punctuated than they are because of the incomplete nature of the fossil record. But both of these are hypotheses that scientists in biology will follow. So gradual change is frequently seen, but there are examples of punctuated patterns in the fossil record that are backed up by experimental evidence. One of those is Helianthus, which is a type of sunflower, Helianthus anomalus, which seems to be a hybridization of two other sunflower species. And this is what it looks like right here. And this is not an example of alloploidy, where we started off with two different species that had different numbers of chromosomes. Here we have relatively closely related species with the same number of chromosomes. In fact, they were close enough, the two chromosomes were similar enough that when you put them together, the hybrid was able to survive. So in an experiment, they took the two species that we thought were the parent species, hybridized them, and in that F1 generation, it turns out you do get some organisms that are viable, but not very many. That said, they found very quick genetic change over time, and after just like 4 or 5 generations of hybrids mating with one another, there was enough genetic change from the first generation of hybrid that what you ended up with looked different from the original hybrid, and actually a lot more like the actual anomalous, right, the actual hybrid species that you see in the wild. So they essentially recreated this in the laboratory and showed that even if you have a hybrid that doesn't work so well, sometimes it can evolve very, very quickly in a short number of generations to get past some of its biggest deficiencies and to survive and thrive. So how long does it take to create a new species? Well, it turns out it varies a lot, from just a few thousand years for some of those cichlids we talked about to 40 million years for some beetles, but it's an average of 6.5 million years. And so this shows you that you can't rely on a speciation clock where, after a certain number of million years, it's time and you make a new species, right? Now you can estimate how long ago there was a common ancestor by the number of species there are, maybe, but it's just an estimate, again, because there's such wild difference between the amount of time that you might find between speciation events. And we also know that while for some speciation events it requires a lot of evolution in terms of change of allele frequency, many different genes have to change in order for these two different species to become reproductively isolated. Sometimes it's just a gene or two. So we've already talked about these Japanese snails that can be left-handed or right-handed in terms of which direction the spiral goes, and that's controlled by a single gene, and left-handed and right-handed snails cannot mate with one another, and so they are a separate species. Similarly, Mimulus, the monkey flower, they have two genes that affect flower color. But flower color influences pollinator preference, and pollinators are how they deliver their genes from one flower to the other, and so that dictates whether or not they can reproduce with one another. And so this couple of genes dictates whether you are this species or that species, and there is not genetic flow between the two. And here's a look at our Mimulus flower. You can see the different colors, and again, this impacts whether they're able to reproduce because it impacts which insect they can get to pollinate. And if you look at this over hundreds of millions of years, you look at the cumulative effects of lots and lots of these small speciation events and extinction events over time, and you get not just different species, but different genera and families and classes and phyla and even different kingdoms and domains. This huge macroevolution is what we're going to be talking about next.